synthesize the food they need to stay alive. This begins with a two-part process known as photosynthesis. The light reactions, or part 1, use radiant energy from the sun to photoelectrically generate ATP and NADPH.
The Calvin cycle, or part 2, uses those products plus CO2 to make a sugar or saccharide from which the plant's food is derived.
Follow along and you will see
how the sugar is made cyclically.
You'll see just how this process is done independently of the sun.
The Calvin cycle will synthesize sugar from carbon dioxide over and over. Spins and spins, finishing when the light begins. The Calvin cycle takes place last in the stroma of the chloroplast, the fluid-filled matrix that's contained outside the thylakoid membrane. The cycle begins initially with a sugar called RUBP, a starting compound which contains 5 carbon atoms in its chain.
The RUBP must bind to carbon dioxide, or CO2, so the plant takes in carbon dioxide from the surrounding air outside. The CO2 is in a gaseous state and diffuses through the leaf stomate, then moves to the stroma where it will be fixated into RUBP.
Rubisco, an anabolic enzyme, helps CO2 and RUBP bind. This is known as carbon fixation, the first phase of sugar formation.
A 6-carbon compound now is formed, but it's unstable and doesn't last long.
It splits in half and generates 2 PGAs, or phosphoglycerates. The Calvin cycle will synthesize sugar from carbon dioxide over and over. Spins and spins, finishing when the light begins. Now I will explain to you PGA reduction, or phase 2.
ATP molecules give phosphate to the PGAs, or phosphoglycerate. Bisphosphoglycerates are produced, which NADPH will reduce.
The bisphosphoglycerates now are changed, and they lose phosphate from the carbon chains. This reaction therefore provides phosphoglyceraldehydes. This is a complex chemical name, so we'll call them PGAL just the same. Or we can call them alternately glyceraldehyde 3-phosphate, or G3P. Several byproducts are also derived as the G3Ps are synthesized. ADP, and it also creates NADP plus and phosphates. All of these can be used when the light reactions take place again. The Calvin cycle will synthesize sugar from carbon dioxide over and over. Spins and spins, finishing when the light begins. Let's see how the G3Ps synthesize. Glucose, a 6-carbon saccharide.
Both of the G3Ps contain 3 carbon atoms in their chains. Count up the carbons, 1 by 1, 6 carbon atoms is the sum.
Those carbon atoms, however, will be utilized to make RUBP. More G3Ps must be obtained to build a 6-carbon glucose chain.
So the Calvin cycle spins back to back 6 times in all, to be exact, yielding in totality 36 carbons via 12 G3Ps.
2 molecules of G3P leave the cycle and bind chemically, yielding glucose E6H12O6 from the carbons that Rubisco fixed.
Now let's take a look at phase 3, the regeneration of RUBP.
The 10 molecules of G3P are recycled enzymatically. They're rearranged and phosphorylated, and RUBP is regenerated.
We're back to where we started from, and the cycle's ready for another run. More G3P will be synthesized when the plant takes in carbon
The
dioxide.
photosynthesis, parts 1 and 2, help make G3P. And when they do, plants can make glucose, their major fuel, and other organic molecules.
The Calvin cycle, it spins and spins, finishing when the light begins. The Calvin cycle, it spins and spins, finishing when the light begins. The Calvin cycle will synthesize sugar from carbon dioxide over.
Finishing when the light begins. Woo! Adios, Marisa!